Accurate diagnosis in complex oncology remains a severe clinical challenge. Soft tissue sarcomas, for example, are notoriously difficult to pin down, leading to misdiagnoses that alter the course of patient survival. The standard tool for identifying and typing these cancers is molecular pathology, most commonly through immunohistochemistry, or IHC. However, IHC relies on monitoring a highly restricted, manually curated set of genes and proteins, known as biomarkers. It captures only a tiny fraction of the cellular activity happening within the sample. The problem is not that modern biopsies lack data. The restriction is computational. Processing the full spectrum of molecular information requires a level of power that traditional database architectures cannot sustain. Forcing oncologists to rely on a limited subset of biomarkers yields an incomplete clinical picture. When the underlying data is fragmented, treatment strategies misfire, directly costing patients their lives. To eliminate this diagnostic guesswork, pathology must expand its scope and analyze the whole transcriptome. The whole transcriptome is the complete, comprehensive range of messenger RNA and microRNA molecules expressed by an organism at any given moment. This visual highlights the scale of the data involved. On the left is the data footprint of IHC. On the right, translating a single tissue biopsy into its whole transcriptome yields billions of disparate, unstructured data points. Classical machine learning classifiers collapse under this weight. The sheer processing power required to automatically categorize this much unstructured data is unmanageable for standard computing models. Harnessing the full breadth of the transcriptome requires a transition away from traditional database architectures. The solution lies in a new data science, AI-powered associative context engines. To understand why traditional genomic databases bottleneck, we have to look at how they structure information. These systems operate on ontological models, rigid hierarchies of subject domains that must be painstakingly constructed and meticulously tweaked by human experts. If these models are not extremely specific to a targeted domain, they fail. Their accuracy degrades in direct proportion to how generalized they become. Traditional databases calculate relationships query by query. New data forces a sweeping query from the top down. Every time a new term is introduced, it triggers a sweeping action, forcing the database to recalculate relationships from scratch. This continuous loop creates exponential demand that overwhelms hardware. A query-by-query architecture cannot scale to the billions of data points required for transcriptome analysis. Analyzing this volume requires a different mathematical approach. The Associative Context Engine, or ACE, bypasses this bottleneck entirely. Instead of executing queries, the engine relies on continuous counting. In this multi-dimensional matrix, numeric values on connecting edges update seamlessly as data drops in. The matrix maintains an ever-present tally of all multinodal relationships. By sustaining this, the engine automatically pre-qualifies search areas. Pre-qualifying the data minimizes the effort required to locate specific correlates, slashing the processing power needed to navigate mass quantities of genomic data. It also removes the requirement for domain experts to manually construct specific ontological queries for every possible scenario. Continuous counting converts the whole transcriptome from an overwhelming flood of raw data into a highly organized, computationally lightweight map. Mapping the data efficiently solves the processing problem, but the engine still needs a way to automatically determine the function of an entirely unknown genomic sequence. The engine solves this by treating microbial genomic sequence data, much of which is operon-based, exactly like unstructured text in human languages. In linguistic AI, a word's function is deduced by proximity to known terms. Its meaning is derived by mapping relationships to surrounding words. This translates to biology. The unknown word acts as a hypothetical protein. Surrounding known words represent established pathways, indicating their collaboration. Utilizing compositional linguistic analysis, the engine automatically deduces the structural and functional context of unknown proteins, bypassing the need for explicit manual tagging. To scale this single sequence mapping across massive multinodal datasets, the engine employs a process called augmented reflection. Augmented reflection relaxes strict contextual constraints. It reads the correlates of correlates, aggregating data across multiple steps to map out broader concept networks. This multi-step aggregation reveals deep correlation potentials across entire datasets that traditional rigid pattern matching systems are blind to. To filter noise, the engine uses differentiating stratification. A tracking matrix tracks matching success and failure rates along sliding scales. Domain experts interact with this matrix, feeding their assertions back into the system. This feedback allows the algorithm to dynamically adjust correlation thresholds. Differentiating stratification ensures the engine is self-adapting. By layering heavy AI processing power with direct human insight, the system continuously improves its own decision-making accuracy. Applying this theoretical architecture to the physical reality of the oncology clinic directly addresses the failure rate in diagnosing soft tissue sarcomas. The associative context engine applies its continuous counting and adaptive learning capabilities to the complete mRNA expression profile of a single patient biopsy. This enables the engine's primary clinical function, multitask inference. Bypassing traditional workflows, the engine processes biological samples simultaneously. The data stream splits into 3 concurrently updating output panels. The first stream catalogs the exact pathway and tissue of origin. Concurrent streams analyze the profile to discriminate between normal and disease states, pinpointing the precise cancer subtype. By eliminating manual modeling and queries, this simultaneous multitasking approach outperforms classical machine learning classifiers in cataloging tissue of origin and disease state. Analyzing billions of data points through associative context engines shifts oncologic pathology away from the inherent guesswork of limited biomarkers. It establishes a comprehensive, highly accurate diagnostic framework that arms clinicians with the exact data they need.